Well, today we have the pleasure of listening to Professor Erin Lavik, who's one of the newest, and I would like to say one of the coolest scientists that are here at Yale. Erin received her bachelor's, master's, and doctoral degree at MIT in material science. And she decided that she actually wanted to do something more medically oriented, so she did a project as a postdoc that is aligned with what her research is today. What she's interested in has been motivated by the fact that there's over 10,000 spinal cord injuries each year. And so she seeks to create polymer scaffolds to promote repair of spinal cords. Now, this work has been recognized highly. In 2003, MIT's Technology Review magazine cited Erin as one of the top 100 young innovators under 35. And last year, Wired magazine indicated that Erin was one of the leading thinkers and doers. So make sure you get her autograph before you leave. One of the things that I think is particularly cool about Erin is not only is she a great scientist, but she's also a playwright. And her plays have been produced both in LA and in New York. So this morning, please help me in welcoming Professor Lavik to the podium.
Cool. So I can put that out of the way. All right, well, thank you very much. It's a real treat to get to do this. It's a little early for me on Saturday morning. Usually I'm still curled up drinking coffee somewhere, so we'll sort of continue that theme while talking. Before I really get started, we have to thank Anissa, because she's the reason this is all happening. She's done all the legwork to really make this happen. And it should also be noted that I got this cute little award, 100 people, innovators and all that hoopla. Anissa was one of the other 100 people, so before we get started. So this morning we're going to talk about tissue engineering, growing organs in a dish. And before we get started with the real science behind it, I have to tell you that this is probably one of the most interdisciplinary fields around. And I've had a lot of wonderful collaborators who've made this work possible. Now, in order to do this work, you need things like antibodies, so you can actually see what's going on in the tissue, and those were gifts of some of my collaborators. There's a group of Scandinavians that are involved in this work. We do a lot of our work actually in pigs to see whether or not the tissue is being recapitulated properly. And the Scandinavians are very good at that. The Young lab is the lab that I collaborate with to do retinal research. The Langer lab is the lab that I came from. Bob Langer really is one of the founders of this field. I've collaborated at Children's Hospital, and the most important thing on this slide is that I now have this absolutely wonderful lab right here that really makes this work possible. And I should point out that Noah here is in my lab, and he is phenomenal, and he's actually going to be doing the demos today. So why do we have tissue engineering? How did this field actually start? Well, it started because surgeons are phenomenal. They've learned how to transplant an awful lot of tissues, but we don't have enough tissues for transplantation. Just a couple numbers to give you a sense of this. In 2000, 72,000 people were on the list with end-stage organ failure, but there are only 23,000 transplants, and that's because of a shortage of tissue, and tissue that matches the transplant recipient. We thought maybe we could just put cells in the place of an organ and maybe it would actually recapitulate the organ, and people tried that, and it turns out in large defects, so unless you have a very small injury, that's not true. You really need something else. And so they came up with this idea of maybe we need scaffolding so that the cells can build new tissue. So tissue engineering basically involves taking some sort of cells, combining them with some kind of scaffolding to give them some sort of structure, and growing those two together so that you hopefully have what they call a neotissue, which you can then transplant. Now, this makes sense, this concept of scaffolding. We use it when we build buildings all the time. And if you look at this, you can see you put up scaffolding, you start to build your building. This is actually the Stata Center. I have this photo because my parking space when I was at MIT was overlooking the construction. It was one of the coolest things about the parking space I had because it was quite a schlep from my lab. But we got to actually see this neat building that Gary designed going up. And so they put up the scaffolding and build these crazy buildings, and then the scaffolding goes away. And that's actually a really important part of tissue engineering, that when you use a scaffold to help direct the cells to make a tissue, you want the scaffold to get out of the way when you're done. And you can make lots and lots of different kinds of scaffolds. These are some of the examples of ones that we use in tissue engineering. And these are all polymers. And we'll talk a little bit about what polymers are, but these are things that we can put in the body, and they'll actually dissolve away as tissue is repaired. So you can have things that involve lots of fibers, or sort of these big porous structures. You can have oriented structures. You can have all sorts of weird architectures. And the architecture of the scaffold is going to be very important to the way the tissue forms. So polymers, the things that we're actually going to be making these scaffolds out of, what are they, first off? I mean, it's very easy to start talking, oh, polymers, they do all these things, plastics, they'll save the world. For anyone who's familiar with the film The Graduate, the guy tells him to go into plastics. Well, I saw that film and followed suit, and actually did go into plastics. Plastics are a kind of polymer. And basically what polymers are is they have building blocks, which are small molecules, and those molecules get linked together. And the way that they're linked together controls the properties. So polymers in the end are actually huge, huge molecules. If my laptop here was a single molecule, was a single part of a polymer, one of those building blocks, the actual polymer chain that we'd build for a normal polymer would more than fill this room. It would be enormous. And we can do some neat things with these polymers. Doop, doop, doop. So some of the polymers that we actually use in tissue engineering, some of the original ones, are based on things like gelatin. And gelatin is basically Jell-O. That's a commercial form of gelatin that you see all the time. So I snarfed this off the Jell-O website there. They have lots of cool pictures. But actually, it turns out that gelatin is a really good scaffold. And it's a natural material. Your body actually makes basically a collagen gel is what we call it, which is gelatin. And it turns out cells will grow on it, and they'll do some neat things. But you all have played with Jell-O. You know, it's sort of, what's the commercial, soft and squishy? Well, we can't always get it to be strong enough to make the kinds of tissues we want. So there's some other materials that we can use. And these are the building blocks. We have something called polylactic acid. And for any of you that have ever gone running and you've talked about lactic acid building up in your muscles, well, lactic acid is a natural product in your body. We can make a polymer then of that lactic acid, and it turns out that's a useful thing for tissue engineering. We take these polymers, they're nice and stiff, we can use them to make scaffolds. And then there's a sister material, polyglycolic acid, that's essentially identical to that. And the neat things about those polymers is that they can dissolve when they're implanted in the body, and those small molecules, those lactic acid molecules and those glycolic acid molecules, are things our body knows how to process. So as it dissolves, our body can get rid of the products, which is really important. Another polymer that was used a lot in tissue engineering was actually nylon. And nylon is, in fact, based on a natural material. Nylon is an attempt to make synthetic silk. It's a hugely successful one. It doesn't degrade very readily in the body, but nonetheless, you can make some neat things out of it. So people started working with that before they were able to make degradable versions of nylon. And my student here, Noah, will show you actually how to make nylon. Now, nylon has a really neat history to it, much neater than a lot of other polymers, which is the other fun thing about it. So nylon consists of two components. So it has this one component that has a little chlorine on it that's not coming out well, and another component. These two ends react with each other. And you basically get a structure that has alternating parts to it. So if you put those two components together in a way so that they don't interact until they get to what we call this interface here, they'll actually get to the interface and they'll start to react with each other to form a polymer. And the polymer becomes a barrier. So you basically get the polymer at the interface and nowhere else in the solution. And what happens is, as you pull out that interface, you get a rope. And it's a really neat reaction. This was actually developed in 1935. And what was happening was, we were having trouble getting silk from the Far East. And so people started saying, well, maybe we need to really be able to make silk because we have issues. We know that we're having political tension in the world. There's some chance we may need to be able to do things like go to war, and we're going to need things like parachutes. And parachutes were made out of silk. If we didn't have silk, we couldn't make parachutes. We were going to have a real problem. So there was a huge push at DuPont to figure out how do we make synthetic silk. And nylon was the outcome of that. It was actually done by a man named Carruthers, who's probably the most brilliant man ever to really step foot in DuPont, at least in my mind. He developed the synthetic rubber. He developed synthetic silk. It's arguable. But nonetheless, he developed synthetic rubber and synthetic silk back when we didn't even really understand what a polymer even was. He really laid down a lot of the important chemistry that we now use. You ready to go there, Noah? All right, so Noah is actually going to do the nylon rope trick. So now we have the polymer formed at the interface, and as he pulls it out, more polymer will form at the interface. So one of the interesting things is these molecules are in solution until they form the polymer, which isn't soluble. That's why it starts to form that interface and come out of solution. There's a lot of little molecules in that small beaker. So Noah is going to be doing this for some time, actually. I don't think he's fully gathered. This is why I'm having Noah do this. I can stand here sipping my coffee in a nice, relaxed Saturday morning kind of way. So we're actually using, and I'm going to check which solvents we're actually using in this. Thank you. So we've got, let's see, what is the actual solution? We snarfed this because we were being, so this is actually I'm really going blind this morning. Hexamine diamine. And then we've got it in sodium hydroxide and distilled water, which is pretty common. You use water as the major solution, and then you add a little bit of base to help the reaction go. And also, there's when the reaction goes, there's chlorine on the one component, and there's hydrogen on the other that react. So you get a little bit of acid. So by having the base in there, that gets the acid out of solution, so the reaction doesn't stop. And then the other one is basically the acid, and I believe it's in methylene chloride. So you pick two components, the solvents, the things that the small molecules are soluble in, that aren't soluble in each other. So you basically set up an interface by doing that. So we have water on the bottom, and then we have an organic solvent that's insoluble with the water on the top. You know, you can actually, if you want, stop and do that afterwards for people. It should be just fine the way it is. So you guys can come up afterwards and see, and you can actually come up afterwards and see nylon getting made. But that gives you the idea of what's going on with it. So polymers, it turns out, besides being useful for scaffolds in general, are really neat things. We can play with the properties pretty intensively. And one of the things we can do with them is that these polymers are these long chains, right? They're like spaghetti. And usually when you just have the polymer sitting there, all of the chains are knotted up like spaghetti. Well, if you take that polymer and you stretch it, those chains, those spaghetti lines, start to line up with each other. But it's not really happy being stretched out like that. So you can take it and stretch it and let it cool down so it's stuck like that. But if you heat it up again so those chains can move, they're going to want to go back to that messy spaghetti sense. It's just like your room. When you clean your room, it doesn't stay clean for very long. It likes to get messy again. And that messiness is what's driving polymers with a lot of their neat properties. So there was a cool paper done by Bob Langer and Andreas Lindenlein. And what they actually did was they took a polymer and they made it into fibers, and then they stretched it out and cooled it. And when they heated it up, the polymer actually started to tie itself, because as it contracted, it formed a knot. And they actually use that now for suture material. And the way it heats up is the body is warmer than room temperature. Room temperature is about 27, 25 degrees Celsius, depending on where you live. Your body is at 37 degrees Celsius. So it heats up when it goes in the body, and it ties knots. This is brilliant for surgeons. They don't have to sit there tying complex knots. They just put the suture material in. And it also means that you could make scaffolds with this, that when they get into the body, they change their shape. For instance, they could be very small, but then when you insert them into the body, they expand and fill out the area inside of the defect. So just to give you a sense of how these polymers can really go through some changes when you heat them up, one of the things we have here is, pretty classic. It's just a soda bottle. In fact, I think we cheated and we're using a water bottle, because it turns out it's a little bit thinner. And this polymer has been stretched out when it was formed into this bottle. So Noah is going to heat it up, and we're going to show you what it was like before it got stretched out and stuck like this. Oop. Ah, there we go.
Not use this to dry your hair.
Again, this is why Noah's doing this, so I can stand over here in a nice, relaxed way. So you can see this bottle starting to shrink. And in fact, pretty soon the label will pretty much fall off. So it'll just keep getting smaller and smaller and smaller. The original form, it's actually a very tiny little piece of polymer that they blow up into the large bottle form that you're used to seeing. You can. You need a really powerful hairdryer. I don't think most of the hairdryers I know will actually get hot enough. But you can do it in boiling water. So you can actually take, if you're careful, take a nice big, pot that you would normally make spaghetti in and put a bottle in it, and it will actually do the same thing. Once the water's at that rolling boil, that's a good 100 degrees C, and that's hot enough to make it contract. Uh-huh. So you mean like the the polystyrene ones, the foamy ones, they're also polymers. They probably won't go to as nice a shape because they use a foaming process to make them. You used to be able to do it really well with margarine tubs because they would go back actually to a cube is what they were because they were pressed out and then they were expanded. The problem with margarine tubs now is they decided to make them dishwasher shape, so it's really hard to get them hot enough to get them to go back to that. We tried that in the lab and it turns out they burn before you actually get to the point of getting them back to the original shape. We had fun actually experimenting with which things we could do this with. So we do this now with this because it's faster, but you can do the same thing with boiling water at home. You can, it's,
Could you remold that to another shape?
if you wanted to, yeah, you could. And actually, back when I was a grad student, when we were sort of playing around with different materials to make things, we would often, for instance, start with just polystyrene containers and melt them down and remold them to something that we were using. Traditionally, this would be an obvious way to recycle, right? That you can shrink these things down and then reuse them. The problem is that a lot of times there's additives that end up getting mixed in during the process, and people who make bottles and other products are very concerned that those additives will affect their bottle. I mean, the last thing you want is a soda bottle that bursts spontaneously, and that tends to be why recycling hasn't caught on quite as much. It's not cheap enough for them to worry about the risk of having impurities in their system. So, Noah, you can stop if you want, or you can just keep going. So Noah's clearly enjoying the thing. Uh-huh. So that's one of the cool things about this. The bottle maintains its shape basically until the end. It'll basically shrink down, and what happens is when they make these bottles is they basically make a little one and expand it till it's bigger. So ultimately it'll stop looking like a bottle, it'll stop having that sort of empty space inside, but in the beginning you'll still have essentially a smaller bottle. So, and again, it works really nicely in the boiling water because it's so uniform the way it heats it up, whereas Noah, while he's very good with his hairdryer, is probably slightly less uniform than the boiling water. So.
What's the process of expanding it? Injection mold or polymer?
Well, what they actually do is it's an injection molding process, so they squirt molten polymer into a mold, and then they actually have a blower that blows through the mold and inflates it. So they call it bottle forming, but basically it's like blowing up a balloon. And once it gets blown up, they cool it down very quickly so it can't go back into its original shape. So it freezes that polymer in that structure. Thanks, Noah. So we're going to move on to the polymers. These polymers are all great, and there's neat things we can do with them, but again, they don't necessarily dissolve in the body. So we've come up with some polymers that do dissolve in the body, and these are based on basically things your body can metabolize. And you'll notice a theme among all of these polymers is that they all have the same structure. They all have a carbon and an oxygen. This is an ester group, and it's these esters that get broken down, that get attacked in the body by water in your body. So you can see these guys all have the same ester groups up there. And actually having your polymer break down by water is really useful because we all have water in our body, and hopefully we all have relatively similar amounts. You can get polymers to break down by enzymes in your body, and that can be useful too, but the downside of that sometimes is people have different concentrations of enzymes, so you can't necessarily count on a particular structure building down in the same way. Now, one of the cool games you can play if you do the enzyme breakdown technique is that if your tissue, as it grows up, makes enzymes that break down the scaffold, you can basically have a very active material that breaks down as the tissue forms in a really tightly controlled way. The chemistry for doing that is tricky, but it's starting to be done now because people are realizing for certain tissues you need the structure to be mechanically strong. And so you need the scaffold to be strong until the tissue is strong, and that's a way to link the two. So we have all these polymers, we can play some games with them. How do we actually go ahead and make a scaffold? Probably the easiest way to do it is this process called salt leaching. And for those of you that think science is, hard and complex, in all honesty, most of it is very simple stuff. And salt leaching is a good example of this. You start out with a polymer solution. So we take our polymer, this can be these degradable polymers, and we dissolve them in a solvent. And what it means when we dissolve it is those long molecules stay as long molecules, but they now get separated from each other. So we end up with a solution. It's just like sugar in water or salt in water, except for instead of being sugar or salt, it's big long chains. And we pour that solution then over salt. And so this is this good old-fashioned rock salt, something you have sitting in your house. What we do, the one trick we play, is that we sieve the salt. So we get salt where the particles are all the same size. We pour the solution over the salt, and we let that solvent evaporate, and that leaves behind a block of polymer with salt crystals sitting in it. We can then just get rid of the salt by soaking this thing in water. That's all we do. We soak it in water. We know that salt is soluble in water. Our polymer is not soluble in water, so the salt dissolves and it leaves behind a polymer with pores in it, and the pores are exactly what the salt was. And this is actually pictures of that. So this is a sponge that we made in my laboratory. So it's a relatively thin disc and it has lots of pores in it. You can see through it. And those pores were created by the salt. And we then went ahead and put this in a scanning electron microscope so that we could look at high resolution and really see what the architecture of the scaffold is. And so this is the scanning electron micrograph, basically a photo in the microscope of it. And you remember, if you've looked at salt, you know that it's cubic. You know, if you look at little table salt, it's always these little tiny cubes. And so our pores are also cubic. So you can kind of see that nice square structure there. So we can make scaffolds. We can use a whole bunch of techniques. This is just one technique that we use in the lab to make a scaffold. It turns out you can do some pretty neat things just with this simple technique. The one thing you don't get typically using this technique is you don't typically get, for instance, long channels. It's hard to get the salt to line up. So we've developed a few other techniques to get different architectures, like those ones you saw way back in the first slide. But nonetheless, if we want to make a scaffold with an architecture, we're pretty much at the point where we can do that. So the next question is, we need cells. The whole point of making a tissue is you take the scaffold, you put on the cells, and hopefully you make a tissue. And what cells are we going to use? So for some tissues, we can use autologous cells, and that means that we use cells from the host. A good example of this is cartilage. If we want to make a new piece of cartilage, and for those of you with achy knees, this is probably sounding really good right now, we can take a scaffold, we can take a little biopsy of your cartilage, and actually they usually do the biopsy from right behind the ear, and we can put those cells, we can expand them because they tend to proliferate, we can get them to divide. So we can get those cells to divide on our scaffold, and now we can have a scaffold with cells that we can reimplant in your knee and help you with, those of you who may have gotten rid of all your cartilage through all those years of running. Sometimes, though, you don't have cells that we can harvest for whatever reason, in which case we can start to think about getting cells from a donor. So we can actually then, just like a transplant, where you instead of transplanting a whole organ, we can isolate those cells from that organ and grow those cells up on the scaffold and transplant it. Now, there are people that are trying to look at actually transplanting cells not from a human, but from a different species. And that may be one option for getting cells. Right now, that doesn't exist, but that is something that scientists are looking at. But there are cases where you don't have cells that we can expand. And a pretty classic one that I work in is neural cells. You probably have heard at some point that neural cells don't divide. When you have a nerve cell, that's pretty much it. There may be a few cells that divide, but probably not a lot of them. In which case, people are starting to look at things like stem cells to see, is there another way that we can get cells that can maybe divide? So this now is that same salt leach scaffold that I showed you before. And what we've actually done is we've covered it with cells. And actually, these particular cells are neural stem cells. These are mouse cells that I actually worked with in my research way back when. And we're able to basically cover our scaffolds with them. So now we have a scaffold with cells on it. So we can really make sort of a neo-tissue. And the question is that tissue going to work? Can we do something with it now that we've made this structure? So some of the tissues that people are looking at to see if they can really make something that's functional are cartilage, and in theory that's probably the easiest tissue to make because cartilage doesn't really have a vascular supply. You don't really have a lot of blood vessels. So you don't, in theory, need to worry about getting lots and lots of blood vessels in the structure. So you can just take the cartilage cells, put them on a scaffold, and implant them. That certainly was the thought. It turns out it's a lot harder to do than that, but people are still working on it. There is a product that's on the market called Carticel, which is by Genzyme, and this doesn't actually really involve a scaffold, but what they do is they take the cells from behind the ear and expand them, and then basically use what's left behind in your knee and re-inject it. And that's working all right. It doesn't tend to necessarily last forever, but for a while it gives you some relief. People are also looking at doing things like heart. making new heart tissue. And this isn't necessarily making an entire heart at this point, but maybe making a heart patch that could basically be put on to help your heart function. And they now actually have scaffolds with heart cells, and heart cells are really cool cells because they beat. So you can actually put these cells on a polymer and they'll contract the polymer just like your heart beats. And those are starting to become a little bit closer to reality. They're not at the point where they can do clinical work with it, but they're at the point where they've got this organized tissue which behaves like a heart tissue. The question is, can they make it big enough to be useful?
How do those tissues get nutrients?
So that's a really good question. In the case of cartilage, they don't need much nutrients, it turns out. They're not a very what we call metabolically active tissue. In the case of a heart, it's a huge issue because your heart is extremely metabolically active. It really needs a lot of nutrients and a lot of transport. What's been found is if the tissue is thin enough, it basically gets transport by diffusion. But when I say thin enough, we're talking, you know, 0.5 millimeter thick, which most of you probably know your tissues are a lot thicker than that. The way your body gets nutrients is through a vascular supply. And that is actually the holy grail right now of tissue engineering, is how do we build a vascular supply into these tissues we're trying to make. And that becomes complex because if we're trying to make a heart tissue, we not only need to get the heart cells in the right place, but we really need to get those vascular parts built in the right formation so that it actually feeds that tissue. And that's why this heart tissue isn't big enough right now to be useful. Liver has a similar issue. Liver, we can actually get the liver cells to organize, but we haven't figured out how to get them enough nutrients to really be a functional liver. Bone is a little bit hard because we need it to be so strong, and that's really been the limitation. We can make new bone tissue, but typically it can't really absorb enough weight to necessarily be transplanted. But one of the things that's come out of, and this is one of the neat things about science, is in doing this we've started to really understand how to integrate at least implants and other structures into bone, because we're learning more about how bone grows. Skin is actually one of the ones that's been probably the most successful, and there's a bunch of different things that have been on the market that have actually been used as scaffolds for skin, either after burns or after you get an injury to your skin or an ulcer on your skin. In all honesty, to give you a sense of how successful tissue engineering is as a field, every company that started a tissue engineering product has sort of gone bankrupt. So we have some issues, and not surprisingly, companies are a little nervous at this point about tissue engineering. Now, two of the things that you can also conceivably tissue engineer, and two things that I work on, so we'll focus on those a little bit, are the retina and the spinal cord. So the retina and the spinal cord are both parts of the nervous system, and you have some basic cells in your nervous system. You have your neurons, these are the nerve cells that carry these impulses, and they have things like axons, which are these long processes. These are these electrical wires that run through your spinal cord and out to your appendages and carry all these signals. You also have supporting cells. For instance, this one, which, by the way, just to give you a sense of the joy of science, I actually had to sit down with a friend and learn how to say this word, and she had to correct me about 10 times before I actually was able to say it. Oligodendrocyte. It's a real fun word to get to say. And this is a neat cell that works in your nervous system to form insulation around those axons. So just like when you have wires in your home, you need them to be insulated so you don't get crosstalk and you don't get zapped. Your body needs insulation as well. So we have cells that make that insulation. Now, looking at the retina, one of the things that happens is there's certain diseases of the retina where you lose your photoreceptors. And these photoreceptors, these are the cells that turn light into electrical signals. They're really important cells. In order for you to see, your photoreceptors have to work. And so one of the interesting things about our eye is the photoreceptors are in the back of the eye. So the retina is along the back of your eyeball, but the photoreceptors are now at the back of the retina. So light has to get all the way through your eye and through your retina to the photoreceptor before it gets turned into an electrical signal. It's probably not the smartest way to do it, but it's the way it happens. So the architecture of your retina is going to be really important. You need your photoreceptors, and you need them to be in the right place to integrate with the rest of the tissue and to work. Now, we have a cell type called a retinal progenitor cell that we know, at least in mice, can sort of form photoreceptors, but it doesn't do it very well. This is one of the big issues. When you hear people talking about things like stem cells and how fabulous they might be, well, we have a long way to go before they're fabulous, because one of the issues is when we transplant these cells, most of them die pretty much right away in most cases. And we haven't figured out how to keep them from dying. So we're not even talking about making new tissue right now. We're talking about keeping the cells alive long enough to even have a chance to make new tissue. But before these cells die, we've seen them a few times form photoreceptors. So we made scaffolds like this, and these are just porous sheets where we actually have channels running through the sheets. And the idea is that if we pour these progenitor cells over these sheets, hopefully they'll line up just like they line up in the eye, and we can slip this sheet behind the retina, and hopefully these cells will integrate with the rest of the tissue. So this now are these sheets seated with these cells. And one of the neat things that we do, we have to know what happens to these cells when we transplant them. So to make them different from the host that we transplant them into, we take these cells from animals that are green so that the cells are green. And we can look for the green cells and find out which cells are the ones we transplanted. So it also makes it easy to get cool pictures. So we put the cells on, and after a few days, these cells grow up really nicely on this scaffold. And actually what we see is that when we transplant this, after 14 days, the cells are still here. This is the first time we saw cells at 14 days after transplantation. Before this, it was only maybe 1 or 2% of the cells that survived. So there's something about giving them a structure that makes them want to survive better. And we're still trying to understand what it is about that structure that's attractive to the cells that they can really survive and actually start integrating with the tissue. And so we wanted to now look at how they integrate. So we take the tissue and we stain it for different markers. And we use markers that normal, healthy cells should express. And the thing that we found was all of these progenitor cells, these magic cells, they're supposed to make photoreceptors, didn't really have any markers for any of the photoreceptors. So you can see, you'll see wherever we have arrows here, that's really where we're seeing these markers for these cells. And there aren't very many arrows, because basically our cells don't seem to want to become photoreceptors in large numbers. A few of them start to express markers, but basically they're all still sitting there. They're surviving, but they're not becoming tissue yet. So we need to figure out how to signal to these cells to actually become photoreceptors. Say to them, okay, now you're in there, everything's great, start to differentiate. That's the term we use for when they start to become the more mature tissue. So we also have done similar kinds of things in the spinal cord. And the spinal cord is one of the things that actually my lab is deeply interested in. Now, the structure of the spinal cord is very complex, but one of the neat things about being an engineer is you don't tend to get too worried about the details. So while there's all these complex tracks that do very special things, as an engineer, I looked at this and said, oh, so it's 2 parts. You have a central section here, which we call the gray matter, and that's where all the cell bodies lie. So that's where all the neurons, basically, and all their supporting cells are. And then you have this outer section, which is called the white matter, because if you actually look at the tissue, it's white, and that's because that's where all those axons are, and the insulation is what makes that tissue look white. So on the outside here, you've got long tracts of axons running up and down through your spinal cord, and on the inside, you've got those cells. Now, Anissa mentioned some of this, but just to remind you, there are 10,000 new injuries each year in the U.S., and the median age in injury is only 26 years old. Medical technology has improved to the point where survival is reasonably good after spinal cord injury, but there really aren't effective treatments that lead to improved functional recovery after the injury. Now, when you have a spinal cord injury, you have a bunch of different things that happens. A lot of times you'll either severage or damage those axons. Again, that's the wires that are running through the spinal cord. You'll lose that insulation on the axons. And as you can imagine, just like in your house, the wires don't work as well when they're not insulated. The same thing happens in the spinal cord. And you'll also just lose the cells entirely. So our question was, could we actually maybe regrow axons and replace the cells that are lost? So again, being a good engineer and seeing the spinal cord as just 2 parts, we basically wanted to create a scaffold that had 2 parts. So the architecture of the scaffold mimics the architecture of the spinal cord. And this is what we came up with. Now, this was going to be done in a hemisection model. So this is where only half of the spinal cord is missing. So we actually, again, this is just a salt leach sponge like you all saw made earlier. And then we took another scaffold that has oriented channels. The salt leach sponge was going to be seeded with neural progenitor cells. So these are like neural stem cells, a little bit more mature than neural stem cells. And again, these are mouse cells that we use. And we use these because we can't just take neurons and put them in there. They won't survive. If you take out neurons and try to get them back in, they'll just die. But the progenitor cells don't die. So at least it gave us a chance to see, can we actually replace cells? And if we do, is it helpful? So we put these progenitor cells on the inside in the salt leach scaffold. And the outside was designed so that hopefully axons could regrow along that. And it's tied together here with a piece of degradable suture material. So if any of you have ever gotten stitches that dissolved, this is that same material. And this dissolving stitch material, suture material, is actually what we're making all of our scaffolds out of. It's the same kind of stuff. So basically, we created an injury. And we either did the treatment with the cells plus the scaffold, or the scaffold alone, or the cells alone, or just the injury, so that we could see what actually happens. And this is an animal from the injury group. So this animal has the injury, but no other treatment other than the standard stuff. So what you can see is it doesn't have weight-bearing stepping. That's what we call it. Its feet are behind it. It can move them, which is really nice, but it can't actually support itself on them and walk in the traditional way. And you can also see that the feet are in this sort of cramped position, and that's very classic after these kinds of injuries. What's that? Okay. Well, the animal's going to motor on for a little bit here. So you can see the animal's also quite curious, as they often are. They're always investigating things. So usually looking for snacks. It turns out one of their favorite foods is Fruit Loops. So this now is the treatment. So it's gotten the scaffold seated with these progenitor cells. And the big difference is that it actually has this weight-bearing stepping. That's what we're really looking for. So it's lifted up now on its hind limbs. Now, one of the other things we look for is to see how those hind limbs coordinate with the front limbs. And it doesn't have very much coordination. So it's far from a perfect animal, but at least it has a major and significant recovery. Now, this is how we try to quantify that. As a scientist, one of the biggest things we have to do is, yeah, we can see something and we can think, great, oh, that's fabulous. But we have to be able to quantify it to know, is the difference actually big or is the difference small? So we use a scale, and it's called the BBB scale. It's named after the people that developed it. And basically, the scale runs from 0 to 21. And what you can see is our scaffold plus cells is basically coming out at a 12 when the animals are fully recovered. So it's far from complete recovery. If it were complete, it would be at 21. But it's got that weight-bearing stepping, which is why it's a 12. Now, the lesion, the one where it just has the injury, and the cells alone are completely identical. So it turns out in our hands, the neural progenitor cells did not improve recovery. What did improve recovery is having a scaffold in place. And actually, even though these two lines look different, the pink line and the green line, mathematically it turns out those lines are essentially the same. So what it comes down to is they're basically, even though we have 4 groups, we really only can see differences, 2 major differences. Either you have a scaffold or you don't have a scaffold. So that means that the polymer scaffold itself is doing a lot to help recovery happen. Now the question is then, well, what's the polymer scaffold doing? If our progenitor cells really aren't contributing to recovery very much, and the polymer is, why? What's actually happening? And what we can do is we can then look at the tissue and understand what's happened to the tissue itself. We know that the tissue correlates with the function. And what we see is in lesion control animals, there's degeneration of the tissue. So the tissue, we remove a section of tissue, and what's actually happening is the tissue around it is falling apart over time. And in fact, the other thing we see is we also often see a lot of scar tissue. So not only is the tissue falling apart, but we're actually getting stuff in there that we don't want. Scar tissue is not going to be functional. When we have the scaffold in place, the tissue doesn't fall apart as much, and actually, instead of having the scar tissue, we often have much more normal tissue in place. One of the things that often happens in the spinal cord after an injury is the tissue starts to degenerate. And there are a lot of different things that are associated with that. The ingrowth of certain kinds of cells is associated with that degeneration, as well as hemorrhaging. And we think our scaffold helps do things like stop the hemorrhaging of the tissue and help inhibit the bad kinds of cells that grow in, those that make scar tissue. And by inhibiting the scar tissue, it creates an environment where the rest of the tissue can actually heal. Now the question is back to these progenitor cells, essentially these stem cells. What are they actually doing? Why didn't they contribute to recovery? They're supposed to be magic cells. Well, it turns out that these cells are basically, they're still there. They've survived to the end of the time course, so we do find the cells, but the cells never actually differentiated. Just like we saw in the retina, the stem cells actually are remaining stem cells. So they're not becoming neurons, and they're not becoming the supporting cells. They're just sort of sitting there. So if we do want to use them, if we do think they're useful, we're going to have to give them some signal, just like in the retina, to get them to actually become mature functional cells. So we still have a lot of work to do, which is unfortunately pretty common in the field, but it keeps us in business. So that's kind of good. Now, I actually have been motoring on here with lots and lots of stuff. I've sort of blitzkrieged you with a lot of data that we do in my lab and in other labs. But I just wanted to take a few minutes and say, why be a scientist or engineer? Because I get asked that a lot. The biggest reason I can see to be a scientist or engineer is I get to play. I mean, instead of being, at a desk just pushing papers, I get to be in the lab and I get to work with my hands and make neat things, either doing the nylon rope trick, which I actually just do to amuse myself most of the time, or getting to make new things. And sometimes when we make these new things, we actually do something that can make the world a better place. And that is the coolest feeling in the world, when you can really do something. A lot of people also ask me, is it hard to be a scientist or engineer? They know that math is hard, science is hard, and sometimes it is. I actually struggled with math. I was not very gifted at it, and I spent a lot of time plugging through things, trying to learn algebra and things like that. But it turns out, if you do the work, you can actually do some pretty nifty things with it. And when I finally did learn it, it was good. I can now do the kinds of math that I need to do the science I need to do. And I get to have a phenomenal time every day. I get to work with amazing students, and I get to work with amazing people like Anissa because of this. So if you have any questions, I would love to take them, either about the work or about being a scientist or anything else. So please feel free to ask.
These studies about mice with lesions over time, in other words, if you get the lesion in the scaffold versus if you get the lesion, wait 2 weeks, and then the scaffold, or wait a month.
Right. So that's an excellent question, because most injuries, the injury happened a long time ago. If we actually want to try to treat a real clinical person, so a patient, we would need to be able to treat someone who'd had an injury possibly a long time ago. We haven't done that work yet, and the reason for it is the best chance of success is right after the injury, when the rest of the tissue is still there. You haven't had all these processes that make the tissue fall apart. Now, we had what we would consider exciting results. The rats walked. But probably what was happening wasn't that we were growing lots of new tissue that was functional. It was that we were preserving tissue and keeping the rest of the tissue from falling apart. So that wouldn't be helpful at this point. But what we did learn was the scaffold itself is a good template. So if we can add the right stuff to the scaffold, for instance, during development, when your spinal cord is growing, there's a lot of chemicals that are released that tell the spinal cord how to grow. We can actually take those chemicals and build them into the scaffold so that they're released just like it's released in development. And our hope is that that'll help signal to the spinal cord to start repairing itself. So that's actually what we're looking at now. If that works, that would be a really neat thing to try then in a more clinically relevant injury, where the injury happened a long time ago.
What kind of injury did you give the rat?
So it's called a hemisection model. And basically what happens is a surgeon goes in. You have to get all the approvals for this and everything. And we worked with a surgeon who was used to doing spinal cord injury. The surgeon actually goes in and does what they call lateral hemisection. So goes down the midline of the spinal cord and goes down for about 4 millimeters and removes a chunk of tissue. And then puts in the scaffold or just closes it up. So it's a very significant injury. A more common injury that a patient would sustain is a contusion injury, so a bruising to the cord. But if you bruise the cord, you get a similar kind of loss of tissue when you're all said and done, unfortunately, right now at least. Well, I'll start there.
Two things that I've seen in the past that I addressed. One was pictures of the ear growing out of the back of a rat.
Quite striking.
Yeah, was that something easier to do because there's not a lot of
Yeah, so that's actually,
cartilage?
right, so that's cartilage, and there's two things about that that are pretty easy. Your ear doesn't sustain a lot of weight, so you don't have to worry about this being a really strong piece of cartilage, whereas in your knee, your cartilage has to be very strong and withstand your whole body weight coming down on it. The other thing about it is that what they were really showing there was if you create a scaffold with a particular kind of structure to it, you'll actually make a tissue with that same structure. So however you carve your scaffold or build your scaffold into a shape, that's the shape of the tissue. I have to say that that image is both interesting and yet sort of disturbing to see a human ear strapped on the back of an animal. But it really does prove the point that if you can make the right kind of scaffold, you can make the right kind of tissue. And in fact, they went in clinically after that. For patients who had burns and had lost pieces of tissue, they were able to recapitulate that tissue. And the reason they did an ear was there was actually a patient who was hoping to get a new ear so they could be, look more normal.
The other thing was one of some science magazines just came out with inkjet printing of tissue, three-dimensional.
Right.
You would like look at that at all.
Yeah, for what I do, it's probably not, for the neural stuff, it's probably not good. One of the things about the cells that I work with, and we mostly work with these neural and retinal progenitor cells, not necessarily to grow new tissue, but we actually use them a lot in vitro to find out if our environments that we create in our polymers are useful. But one of the things we've learned is these cells don't like to be basically sheared, so they don't like to go through small needles and things like that. So the inkjet printing basically kills our particular cells. For other cells that are a little bit tougher, it might work well, and it might be a really neat way to say, okay, if we're going to build a vasculature, let's put the cells of the vasculature down right here, and then right next to it, we'll put the liver cells down, and maybe we can actually print basically a liver, which, by the way, I have some liver up here, which I sort of forgot to point out, but I made poor Anissa rather ill before we all started with my various liver parts that are hanging out. These are actually chicken livers, so that I got from the grocery store on the way over this morning. So you had a question?
My impression of the gray matter was always on the spinal side, that the axons must be run vertically up and down, and your matrix kind of goes outwards. I'm wondering how you...
So the outer part of the matrix goes up and down once it's implanted. The inner part basically goes out. And that's because the cells that are in the inner part have to be able to reach to the outer part to do things like myelinate the cells. And actually you do have what we call interneuronal connections. You'll have a neuron in the spinal cord that connects with one from the brain. And so we wanted to be able to allow those pathways to happen.
Getting back to the mouse and the ear, was the idea that ear could then be taken from the mouse and attached to the person that needed an ear, or
So
the idea there?
the idea there was actually when they started that experiment, one of the big questions was, okay, you can create a polymer scaffold and you can put cells on it. Will it actually, if you do that, once it's implanted, will it stay looking like the scaffold? Will it still have that structure, or will it over time reform? There was some concern as the scaffold degrades, maybe the tissue, instead of looking like an ear, would form a ball. And the last thing you wanted to do was to, implant that on someone who'd already been through a pretty traumatic injury. So they tried it on the animal to see if it stayed like an ear, because we really had no data on it. It turns out, if it does stay like an ear, you do maintain that architecture even after the scaffold's completely gone. And once they knew that, they were then able to do the same thing on a patient.
Well, why don't we have some questions? Well, why don't we tie this up so those who have other questions can come and talk to Erin. But let's thank her for giving such a wonderful talk.
Thank you. Was it okay?